Good morning, everyone, or good afternoon, depending on what time zone you are in. Good evening. Hello, and welcome to the session, Immune Modulation by Nanomedicines, New Therapeutic Approaches, Adverse Phenomena, and Safety Testing. My name is Marina Dobrevaskaya, and I'm honored to co-chair this session with Professor János Szebeni of Semmelweis University. Before we start, we have few housekeeping items, announcements to make. We would like to please ask everyone to mute your microphones unless you are presenting. This is to help us with the bandwidth. And if you have any questions, please type your questions in the chat box. Throughout the sessions, if the speakers finish earlier than the allocated time, then we will have a chance to ask questions immediately after individual presentations. But we will have also discussion at the end. So if we don't have time to address questions right after the presentation, we can save these questions and ask them at the end. And János and I will take turns in announcing speakers and reading your questions to speakers. And with this, it is my pleasure to announce our first speaker, Professor János Szebeni of Semmelweis University, and also co-chair of this session. And he will talk about a very important subject in the world of nanomedicine. He will talk about the issue of infusion reactions and how to assess infusion reactions to nanomedicines. So János, please, the microphone is yours.
In my talk, I am going to focus on one critical safety barrier in nanomedicine, that is to say infusion reactions, talking about the models, mechanism, future directions. And as you can see, I am coming from Budapest, Hungary, Semmelweis University, where we have a nanomedicine section in the Institute of Translational Medicine. And also I am heading a little company, immune toxicology service company called Seroscience. This is an outline of what I'd like to present today shortly about infusion reactions, its features, just to remind people who are not initiated. Then talk about some classifications of it. There are many classifications: symptoms, mechanism, models, prevention, and some recent progress experimentally. Namely, we have been using TICs for immunogenicity assessment, and also some recent conceptual development regarding the role of complement and the use of the pig model. So just going rapidly through the basic features of reactions. It is an acute allergy syndrome. There is huge individual variation of the symptoms. It can be severe, more or less. It can also be fatal. It is associated with intravenous therapies. It is caused by nanodrugs and other drugs, but also biologicals. And this aspect is even important because biologicals now represent the state of the art. These reactions arise at therapeutic doses. Usually they arise after the first infusion or injection. They are relatively rare, although they can be quite frequent. There is a phenomenon called tachyphylaxis, which means opposite to a traditional allergy, these reactions can self-mute, their self-tolerance can develop, which is used for their prevention. It is a cascadic immune reaction with multiple redundant pathways. All arms of the immune system takes part in it, innate, specific, humoral, cellular. And unfortunately, it cannot be predicted by a standard allergy test, and that makes it more difficult to handle. It it means a hurdle for the translation of nanotechnology-based drug products, sometimes, not always, of course, as it may lead to drug withdrawal. And regulatory authorities increasingly demand preclinical testing. We had the fortune to review the subject 2 years ago with Marina in a Nature Nanotechnology paper, as you can see here. This shows that infusion reaction actually is a problem that exists ever since pharmacotherapy is applied in man. And it should be a medical historian to trace back the first description of such a reaction. But definitely it has been present ever since something is injected directly to the blood. Here you can see the symptoms on the left bottom, mild, moderate, severe, nausea. I would not read all this, you can have a glance. What I'd like to more highlight that there are many different ways by infusion reactions have been classified over a long time of research. The symptoms can be classified according to the organ systems they are impacting. According to severity, I have already mentioned the mild, moderate, severe, but there are other ways to classify infusion reactions. The best known is perhaps the oldest, originating from the 70s, which grades these reactions from 1 to 4. Reactions can be also classified by their kinetics. According to this, they can be acute or delayed. The prevalence can be very low, moderate, very high. Duration can be short or extended. Actually, you can see all different manifestations of the same phenomenon. And the mechanism has traditionally been assigned to the so-called type 1 reactions, according to Coombs and Gell's classification. But this recently, the type 1 reactions have also been dissected to different arms. This is a list of drugs causing such infusion reactions. I wouldn't go into details here. The bottom line is that different types of drugs can cause it, including liposomal drugs, micellar drugs, monoclonal antibodies, pegylated nanoparticles, contrast media. This table here shows the pegylated nanodrug being shown to cause such reactions. And as you can see in the bottom with black background, some of them have already been withdrawn from the market because of such reactions. Here is an interesting calculation about the economic impact of such reactions calculated for satellizumab. It shows that if the infusion reaction occurs in a hospital setting, it almost doubles the expenses spent on the drug. Here from many thousands of dollars increase what has been calculated. The mechanism. Well, from the beginning, it was proposed that the reactions are due to complement activation. Complement is part of the immune system, and a large fraction of these hypersensitivity reactions could be associated with complement activation in experimental setups and also in clinical studies. So the CARPA means complement activation related pseudo-allergy. It is not a real allergy, pseudo-allergy, because of the absence of IgE mediation. And this CARPA concept has been emphasized and used for more than 20 years. But recently, it had to be a little bit revised on the basis of some debate in the literature on its involvement in these reactions. Recently, nevertheless, a very stunning evidence was published about the role of complement activation in these reactions. Here is doxyl or doxysome, another form of doxyl, has been known to cause such reactions, and it has been shown in vitro that if the doxyliposomes are incubated with antibodies specific against the PEG on the vesicles, then the vesicles open up, their content as a consequence of complement activation, and this has been visualized here as evidence of the membrane attack complex. So there's no doubt that complement can be activated, and it can cause harm to the drug, but also the liberated anaphylotoxins, as you can see here from the mechanism, it also can harm the host. And what is happening? Well, these are some glimpses of the animal models. Complement is activated by the drug, and then the anaphylotoxins C3A, C5A, maybe C4A accumulates in blood. And when it reaches a certain threshold, it starts to activate cells which are responsible for allergic reactions. Here a mast cell is presented, although macrophages can also do it. And the complement, via the complement anaphylotoxin receptors, these cells are activated and release a lot of mediators which then cause the symptoms. Now, with time, we realize that it is not only complement which contributes to the reaction, but also there is a direct impact on these cells by the nanoparticles, as illustrated here. The second hit, so we are referring to the double hit mechanism of hypersensitive reactions. But it turns out that maybe even more hits are needed together, which is illustrated here. The complement activation can activate allergy-mediating cells, not only through anaphylotoxin receptors here, but also by other receptors, including C1q receptor, MBL receptor, receptors for C5b9, the membrane attack complex also has a receptor. So there are many different ways by which these cells can be activated, and we refer it all as CARPA because complement mediated. But it turns out that there are reactions which are not complement mediated. We refer to them complement independent pseudoallergy or SIPA, which can be mediated by Fc gamma receptors and other receptors. So overall, it's a very complex phenomenon. No wonder why there is such a tremendous individual variation. In vitro tests for CARPA prediction, obviously because of the role of complement, there is the most obvious assay is to test for complement activation in sera, and there are many animal and human studies which attest to the relevance of such tests. The proof of concept of testing complement in vitro in sera has been quite strong. And you can see on the right side what kind of assays do we have for human complement mediators, also for pigs, rats, mouse. So we have quite a few different approaches how to assess complement activation in experimental animals and in man. About the animal models, the phenomenon being a very basic phenomenon is observed in many different species, although in mouse, rats, the reaction is much less sensitive to the drug than other models or human. So the bottom line is, or the overall message, that these species are not good for predicting or testing or studying CARPA. Dogs do show the reaction, but it is more difficult to experiment with dogs and also non-human primates than with pigs. Our focus today and for the past 20 years, we have been studying pigs as a model for CARPA. And here is the basic phenomenon. Administration of a small amount of nanodrug causes major hypertension or in animals, which is associated with a significant rise of pulmonary arterial pressure, arrhythmias, skin flushing, which mimics the human reaction, and a lot of other changes which we are measured. Quite a few of papers using these the pig assay. Here's a feature of the pig reaction, namely that hemodynamic response of pigs is quantitative and specific, and it is also a very perhaps in the future a useful feature because it may enable to immune imaging the immunological properties of nanoparticles. Here are nanoparticles which are injected in pigs. You can see the pulmonary and systemic reaction. Each different nanoparticle, the pattern is different. And it is typical of the nanoparticle, typical of the dose and the administration method. So if everything is standardized, that could serve as a fingerprint for the nanomedicine immunological activity. Another evidence for the specificity and sensitivity of the assay, we have found that pigs enable to differentiate between different nanoparticles according to their size. It could differentiate spheres, rods, discs, etc., which is an important property. In lack of time, I wouldn't. I just tell you what interesting new developments have been described recently. First of all, the pigs allow not only the immunogenicity of nanoparticles, and evidence for it has been for regulated nanoparticles. Pigs can be used to develop safe infusion protocols. It has been used for the FDA approval of Onpattro. This is a patent by Alnylam. They used a pig model to develop a safe administration protocol. Another recent paper describes the development of safe administration for regulated liposomal prednisolone phosphate. But Metzeler yesterday mentioned that infusion reactions were an unsolved problems for this drug. So pigs may play a role in solving this. And here is a scheme, a testing scheme, which enables to predict the reactogenicity and immunogenicity of nanoparticles. It was endorsed in a recent article on proposed strategies for immune toxicology testing by regulatory agencies, and they highlight the fact that the current protocol, the current guideline, the ICS-8, does not test which these reactions. There's a prevention which has been worked out recently. This is a paper with Dr. Gabizon, which details the various drugs that can be administered for pre-medication for the case of liposomes. And this is my last slide to highlight that there is a conceptual progress in these reactions, in that we have to emphasize more that it is not only complement but also other factors, and the pig is risk identification model and not a toxicity model. It serves to identify the risk of corpora and other side effects. And with this, I'd like to thank your attention and thank my colleagues who participated in this work for many years now. Thank you very much.
Thank you, János, for the great presentation. Since we are running a little bit over time, I suggest that we reserve the questions to our speaker till the end of this session, and we continue.
Okay, so then it is my pleasure to introduce you, Marina, as next speaker. Just a sec. I have to, did I step out from sharing? Yeah, okay. So Marina Dobrevaskaya is head of the Immunology section at the Nanotechnology Characterization Laboratory in Frederick, which belongs to the National Cancer Institute. She is also an associate of Leidos Biomedical Research, and she is probably, or for sure, one of the best recognized experts in our field. So Marina, the floor is yours.
One more time, hello everyone, and thank you for joining, and thank you, János, for the introduction. While I'm waiting for the presentation to load in the presentation mode, I would like to say thank you to our technical IT support, Heinz, who is helping us to run this session. I want to start my presentation by acknowledging my colleagues at the Nanotechnology Characterization Lab, and especially my immunology team, both current members, Betty, Ed, and team, and former members, Anna, Jamie, Park, and Pink, and Ankit, for their hard work. They developed protocols, and for conducting their studies, they helped us to improve our understanding of nanoparticle interactions with the immune system. This is an outline of my presentation. I'm going very briefly, I'm going to talk about regulatory landscape and general principles of assessing nanoparticle immunotoxicity. I will share with you our experiences at the NCL and provide my opinion about what I think waits for us in the next 5 to 10 years in nanoparticle immunotoxicity evaluation. So immunotoxicity is a scary thing because we have data now showing that clinical studies are indeed can be held due to immunotoxicities. We also know that in the past decade, 10 to 20% of drugs were withdrawn from clinical use due to immunotoxicity. And János shared some of the examples in his presentation. We also know that development of nanomedicines, since these are very complex drug products, it takes a long time and is very expensive. For example, Alnilam's Onpattro product, which is liposomal siRNA, took 20 years and $2.5 billion to get into clinic. And because of this, evaluation of immunotoxicity, understanding where it is coming from early in the product development is very important, not only to create safe drugs, but also to help companies avoid expensive failures later in the product life cycle. Regulatory landscape of immunotoxicity includes ICH guidances, specifically ICH S6 and ICH S8. Those are adopted by many regulatory agencies worldwide. Here in the US, we have a new draft guidance document that was released by the FDA earlier this year. Once finalized, it will represent the agency's current thinking about immunotoxicity evaluation, and it is intended to supplement several guidance documents, including ICH-S6 and ICH-S8 documents. According to these guidances, toxicity to the immune system encompasses a variety of adverse effects, which include suppression and enhancement of the immune response. Suppression may make hosts more susceptible to infections in tumors, while enhancement leads to exaggeration of autoimmunity and hypersensitivity. Both immunosuppression and enhancement can be associated with two distinct groups of drug products. First is the group in which drugs are intended to affect immune function for therapeutic purposes, for example, those to prevent organ transplant rejection, and in this case, immunotoxicity would be considered as exaggerated pharmacodynamics. And the second one are the drugs that are not intended to affect immune function, but they do so through a variety of effects, for example, by causing apoptosis of immune cells. There is a third type immunotoxicity that was not recognized until very recently, and it is immunomodulation. Immunomodulatory drugs modify immune responses. They are not overtly immunosuppressive or immunostimulatory. They may have subtle or even mixed effects, and it is very difficult, it's very challenging to detect this type of toxicity without an additional challenge. So what we know currently from our experiences and from data in the literature is that nanoparticles can be immunosuppressive, they could be immunostimulatory, and they also could be immunomodulatory. And these effects could be due to either API or due to the nanocarrier itself, or a combination of the carrier and API. And in the next few slides, I will share with you experiences of our lab assessing these different types of immunotoxicities. Very briefly, our lab is dedicated to cancer research, but temporarily we are open to COVID-19 applications. The characterization that we do is free of charge for submitting investigators and includes comprehensive physicochemical characterization and in vitro and in vivo immunology, pharmacology, and toxicology. And if you would like to learn more about work that we do, please feel free to contact us. Very first thing that we do, then we start our assay cascade characterization, is we assess nanoparticles for the presence of innate immunity modulating impurities, which includes live microbes, mycoplasma, yeast, bacteria, mold, and also microbial byproducts, endotoxins and beta-glucans. And throughout my presentation, I will include this blue box where I list the assays in our assay cascade that we use to characterize certain properties, and the link to our online page from which you can look for these assays and download full text to learn more technical details about them. Very briefly, we found that about 30% of preclinical formulations fail due to endotoxin contamination, and very common sources of contamination include water and synthetic process. We also observed that about 5% on average fail due to bacterial contamination, and the common sources of bacterial contamination include water, dust, and handling. And you see that there are some years where we see peak in microbial contamination in nanoformulation, and usually these peaks coincide with the time then our sponsors start experimenting with new materials and explore new technologies to create nanomedicines. And obviously, these are very complex products, and there are a lot of challenges that researchers have to overcome. If we look at the types of bacteria that are found in nanoparticle contamination, then one thing stands out. That is, most of these bacteria are aquatic and soil species, which means that environment in the lab and cleanliness in the lab is very important for production of contamination-free materials. Another thing stands out is that some of these bacteria are part of normal human microbiota that lives on the skin and nasal and oral cavity. Therefore, wearing personal protective equipment is very important, not only to protect employees from potentially hazardous effects of nanomaterials, but also to protect nanomaterials from contaminating them with our microflora. I mentioned that NCL is dedicated to cancer research. Therefore, it is not surprising that majority of the formulations that come through our assay cascade are immunosuppressive due to the drug that these materials deliver. However, we did come across a small proportion, about 8% of formulations that are immunosuppressive due to the carrier itself. We are using in vitro assays to characterize these properties, which include leukocyte proliferative responses towards antigens and mitogens, the bone marrow toxicity, and we also have in vivo assays the T-cell dependent antibody response assay to assess this toxicity. In this case study, I would like to highlight one of the examples where a nanoparticle is immunosuppressive due to the carrier itself. And this study was conducted by Ankit in our lab. He worked with Ferrahim, and for most people in the immunology field, Ferrahim is known for its ability to activate macrophages and complement system, but we were interested in understanding how these particles may affect adaptive immunity and what effects they may have on lymphocytes. And so what Ankin found is that exposure of the primary human T cells to Ferrahim suppresses their proliferative responses and cytokine responses that are shown on this graph upon subsequent activation with a stimulus that activates the cells. In this case, it was CD3 antibodies. And he also found that this toxicity is due to the induction of mitochondrial loss, and there is a dramatic change in the exposed to particle cells in the mitochondrial shapes. Here you see damaged and mitoffusion, where you see this huge mitochondria compared to the normal crystal and normal shape of mitochondria in control cells. Immunostimulation is opposite effect to immunosuppression, and it comes in many flavors. Many markers exist, but in this presentation, I chose to speak about cytokines because they are reliable biomarkers. They give a lot of information, and also there is a good in vitro and vivo correlation. It is very important to note that cytokine response depends on the type of nanomaterials and the type of the drug that they carry. What we commonly see, for example, that lipid nanocarriers and liposomes induce chemokines, MCP1, MCP2, IL-8, MIP1 alpha, MIP1 beta, RANTES, while nanoparticles that carry therapeutic nucleic acids or nanoparticles made of therapeutic nucleic acids commonly induce type 1 and type 3 interferons. In collaboration with Hezi, we wanted to understand how particles could be controlled, how particle properties could be controlled in order to decrease their immunostimulatory activities. And we found that ion content of liposome in the liposomal cavity contributes to the immunostimulation. Here you see that liposomes with ammonium sulfates in their cavity are more pro-inflammatory than their counterparts with calcium acetate. In collaboration with Kirill, we investigated structure-activity relationship in response to nucleic acid nanoparticles. And Kirill presents later in this session, so I don't want to steal his thunder. I'll just mention that we found that material types like RNA but not DNA makes these materials more pro-inflammatory, and their shape contributes in that 3D particles are more pro-inflammatory than their planar and fibrous counterparts. Now let me speak a few minutes to speak about immunomodulation. Immunomodulation is a difficult toxicity to catch. And the reason why it is difficult to catch is because a change is required to detect this type of toxicity. And on this slide, I'm sharing with you the results of the study that were conducted by Anna in our lab, where she looked at the procoagulant activity. Procoagulant activity is a phospholipid protein complex expressed on the surface of the activated white blood cells that serves to activate plasma coagulation cascade that leads to the blood clot. And inflammatory stimulus like bacterial lipopolysaccharide at high concentration have the ability to induce procoagulant activity. However, at low concentrations, LPS not induce this activity, so as a variety of common dendrimers that we tested regardless of their surface charge. However, if we take this low concentration of LPS and we treat cells with this concentration after they were exposed to dendrimers with cationic surfaces, in this case we're using amine-terminated and guanidine-terminated dendrimers, then we start seeing procoagulant activity. So in other words, exposure to nanoparticles sensitizes cells to produce or strongly react to another pro-inflammatory stimulus, which otherwise cells do not respond to. There are other common types of reactions to nanomaterials. They include hemolysis, complement activation, and János spoke extensively about this in his presentation. Change in plasma coagulation time, platelet aggregation, leukocyte procoagulant activity. And in our lab, we assess them using a variety of protocols. And here we see a summary of structure-activity relationship. Many of these I mentioned in the previous slide, so I will proceed to the next portion of my talk, which is outlook into the future. As a community, where do we go in the next 5 to 10 years to improve our understanding of nanoparticle interactions with the immune system? Well, in my opinion, we as a community have to work together to improve current and establish new approaches to study a variety of effects. Specifically, we need to better understand contamination of nanoparticles with innate immunity modulating impurities. Endotoxin is not alone. Beta-glucans, other innate immunity modulating impurities can contribute to nanoparticle immunotoxicity. And we need both good methods to reliably detect this contamination and understand the consequences of this contamination to the safety of nanomedicines. János emphasized it's hard to underestimate the importance of understanding the mechanisms underlying infusion reactions to nanomedicines, and most importantly, identifying reliable models, both in vitro and in vivo models. I shared with you some examples of the signature cytokines that are biomarkers of inflammation to certain types of nanomaterials. And we need to expand and understand beyond liposomes, beyond nucleic acid nanoparticles, and also what can be used, what strategies could be used to decrease this immunostimulation. Immunogenicity of APIs, platforms, and components is another important topic. Antibiotic antibodies can contribute to enhanced complement activation by pegylated nanomaterials, as I show here from in vitro study. And now we have increasing data about pre-existing antibodies and ability of certain nanomedicines to induce antibiotic antibodies. Therefore, more studies are required in order to fully understand this phenomenon. Very new area, and this is where I think the future of nanomedicine is to understand long-term toxicity of nanomaterials and specifically understand the autoinflammatory reactions and autoimmune responses to nanoparticles. These studies take time and they take special models, and so a lot of research is ongoing in this direction currently. And last but not least, mechanistic toxicology is important because only then we understand the mechanisms of nanoparticle immunotoxicity. Not only we can design safe nanomedicines, but we can also use this information to repurpose or redirect this toxicity to beneficial purposes. So thank you very much for your attention. And I also ran out of my time, so we will proceed with the next speaker. So János, please take the microphone while I am unsharing my screen.
Okay, do you hear me?
Yep.
Okay, the next speaker is Dr. Joke Han, who's an associate professor at the Department of Molecular Cell Biology and Immunology in Amsterdam. And she is going to talk about splenic uptake of ganglioside-containing liposomes. And more details is coming. Joke, this is your time.
Okay, thank you. I will share my screen. Let's see. Do you see my screen? Okay, well, I want to thank the organizers for the opportunity to present our data. And we have studies on ganglioside-containing liposomes that target to CD169 positive macrophages in the spleen and that elicit strong immune responses. Okay, I'll have to go to the next slides. Oh yeah, sorry, I went too fast. So, sorry, I have to go back. So these CD169 positive macrophages in which we are interested, you can see them over here. This is a section of a mouse spleen on the left, and you see these cells in green located as rings on top of the blue B cell follicles. And this is a schematic overview of the spleen. As you all know, the spleen has multiple functions. The red pulp macrophages remove damaged red blood cells, and in the white pulp, immune responses are being raised against pathogens present in the blood. And in the area in between the white and the red pulp, this area is called the marginal zone. That's over here. And over there, small blood vessels end in sinuses and the blood flow slows down. And then two types of macrophages filter the blood for pathogens. And one of these macrophages is more located towards the red pulp and expresses the cyano-1 molecule. And then the other macrophage is located to the inner side, to the white pulp, on top of the B cells. And this one expresses this marker CD169. And this marker is also known as cyclic-1 or cyanoadhesin. And as I said, these two types of macrophages filter the blood. They remove pathogens, so they remove bacteria, viruses, dead cells, exosome, etc., especially particulate pathogens. But we were interested whether these macrophages have other functions beside their innate functions, and especially whether they can play a role in the activation of the adaptive immune response, so the T and the B cell response. And we have been investigating this CD169 positive macrophage by antigen targeting using antibodies and later on by liposomes. And what we have observed in these studies, when we target antigens using antibody-antigen conjugates to these macrophages, that they can elicit really strong B cell responses. And these are germinal center B cell responses that are isotype switch and high affinity. But furthermore, what we observed is that they transfer antigens to dendritic cells, and these dendritic cells in turn stimulate and activate T cell responses, especially CD8 and CD4 T cell responses. So these macrophages have a really efficient collaboration with these dendritic cells, and I think this is the physiological function of these macrophages. Our ultimate goal is to design vaccines for cancer. And normally we would think to target these vaccines to these dendritic cells. However, with this knowledge in the back of our mind, we thought maybe it would be better to target vaccines to these CD169 positive macrophages because they have this excellent capacity to filter the blood. So this is what we are doing. So our goal is to design cancer vaccines. As you all know, lots of cancer patients are nowadays being treated with these checkpoint inhibitors. However, still the majority of these patients do not respond really well to these checkpoint inhibitors, and this is because they don't have an existing immune response. However, we think if we can combine these checkpoint inhibitors with a vaccine to elicit new T cells, then more patients will respond to these checkpoint inhibitors, make an immune response and attack the tumor. Well, for cancer vaccines, as you all know, vaccines should contain tumor antigens as well as adjuvant to activate the immune system. But we think we should add another third component, and this component should lead to an increased uptake by the CD169 positive macrophages. And we chose to include gangliosides, and I'll tell you in the next slide. So we chose to make liposomes, including gangliosides. Because gangliosides are known ligands for this molecule on these macrophages. And this has already been discovered 30 years ago by Paul Crocker, who identified this molecule CD169. And CD169 or cyclic-1 binds to sialylated glycoproteins and glycolipids. And a number of these glycolipids are gangliosides. And a few years ago, several different groups discovered that cyclic-1 or CD169 binds to different types of viruses because they bind to the gangliosides in the membrane of these viruses. So our approach was to make liposomes, including ganglioside, as a kind of virus-like liposome. So I want to show you two sets of data. One are mouse studies, where we do in vivo experiments, and another set of data is with in vitro experiments using human cells. So to start with the mouse, we made three types of liposomes, one with GM3, which is a binder of CD169, and one with GM1, which is in the mouse system, not a binder for CD169. We encapsulated ovalbumin protein in this liposome, and we performed in vitro, but also in vivo immunization studies with after intravenous immunization. And we added a separate adjuvant, poly-IC and anti-CD40. Well, these are the in vitro studies. If we incorporate different mole percentages of GM3, then you can see over here in an ELISA setting that it leads to specific binding to these CD169 molecules and not to a mutant version of the molecule. if we incubate fluorescently labeled liposomes containing different amounts of GM3 with splenocytes, containing different types of cells, then you can see that the liposomes without GM3 don't bind any cell types, but with the inclusion of GM3, and especially 3 to 5% GM3, leads to binding to CD169 positive macrophages, but not to other cell types. Then we injected liposomes into mice, and we analyzed the uptake and binding 2 hours after immunization. And we first wanted to do an unbiased approach, so we did a TSNE analysis. And as you can see over here, there is a population of cells which take up these liposomes, but they also take up the liposomes without any GM3 incorporation. And if we gate back with known markers for the different types of cells, then we indeed find out that this is the CD169 positive macrophage population. And if you look at the graph belonging to this data, then you can see over here that indeed the non-modified liposomes are already taken up by these macrophages, but that the inclusion of GM3 further enhances the uptake by these macrophages. We don't see much other uptake, but we do see some uptake by red pulp macrophages, which goes down if we include the gangliosides. Well, so this is the uptake and binding, but are these liposomes also immunogenic? So we immunized mice in the presence of a separate adjuvant, and it's a really strong adjuvant. And then after 7 days, we determined the T and the B cell responses. So the T and the B cell responses specific for ovalbumin, which was encapsulated in these liposomes. And because we already saw non-specific uptake by these liposomes, we were already expecting this, that also already the control liposomes and the liposome with GM1, which does not bind to CD169, are already immunogenic. They do elicit already CD8 T cell responses and CD4 T cell responses, but the incorporation of GM3 further enhances the uptake and the immunogenicity of these liposomes. And we have 2 readouts for OVA-specific CD8 T cell responses, and 1 for CD4, and especially the CD8 T cell responses are enhanced by inclusion of GM3. And the same holds true if you look at the B cell responses, then we see an increase in the OVA-specific germinal center B cell response and the antibody titers in the serum of these mice. So from these experiments, we conclude that the control liposomes are already immunogenic, but the addition of GM3 enhances the immunogenicity of these liposomes. Then I want to show you some data from our in vitro studies with human cells. So in these studies, we incorporated 5 different liposomes. And I have to say that here, the GM1 liposomes do bind to CD169. And we incorporated 2 tumor antigens as a peptide and 2 toll-like receptor ligands, or toll-like receptor ligand 4 and 7/8, as an adjuvant. And we first use monocyte-derived dendritic cells, and if you incubate them with type 1 interferon, they upregulate this marker CD169. And if you then incubate them with this fluorescently labeled liposomes, you see binding at 4 degrees, and also uptake at 37 degrees. And this binding and uptake can really be blocked by the blocking antibody. So that's really specific. And it's also very interesting because these modices express other cyclex as well, which are able to bind to sialylated glycolipids. But apparently these ganglioside liposomes are very specific. If you use imaging flow cytometry, you see uptake and internalization of these ganglioside liposomes. Then we used tumor antigen containing liposomes. So we have the Wilms tumor 1 antigen and a GP100 antigen. And we use MPLA liposomes, and we fed them to these modices. And then these modices were incubated with T cells specific for the WT1 or the GP100 tumor antigens. And we detected the T cell activation by the production of interferon gamma. And as you can see over here, all these different types of ganglioside-containing liposomes induced activation of the T cells. And this was with a short peptide, but also with a longer GP100 peptide, which has to be processed. We see production of interferon gamma, and it's similar. The T cell activation is similar compared to targeting to DC sign, which is another marker on MODCs. Well, MODCs are a really nice system, but they're also a kind of artificial dendritic cells, and these types of dendritic cells are not being found in vivo. If you look in PBMC, for example, in humans, then you can find different types of dendritic cells. So it's known that you can find the CDC1, the plasmacytoid DC, and the CDC2. 3 years ago, there were 3 different papers which claimed that there is another type of DC subset, and it's called DC5 or ASDCs. And this DC was characterized by the markers AXL and Cyclex6. And we looked in the PBMC of healthy individuals, but also of cancer patients, and we indeed find this AXL positive DC. And this DC is interesting for us because it expresses this marker CD169. So we set out to investigate whether these dendritic cells can bind our ganglioside liposomes. And indeed, when we incubate the fluorescently labeled liposomes with these ASDCs, they bind, except for the GD3, which is not a good binder for Cyclic1. And again, we can block the binding of these liposomes by our blocking antibody, which shows the specificity of the interaction. And finally, we made a DC prep from PBMC, which is enriched for this AXL DC because it's a not very frequent DC, so it's really hard to sort it out. And we fed liposomes containing the WT1 epitope and the Tol7/8 agonist. And we incubated them with the WT1 specific T cells and looked for interferon gamma production. And again, we could show that the inclusion of GM3 in the liposomes led to enhanced T cell activation. So also we find with ex vivo isolated DCs that these liposomes are immunogenic and stimulate T cell responses. So I come to the end of my talk. I hope that I have convinced you and by our data that shows that the inclusion of the gangliosides in the liposomes enhances binding by CD169 expressing antigen presenting cells, both in mouse as well as human systems, and leads to enhanced T cell activation. And we think that this is a promising approach for vaccine development for anti-cancer vaccines. And finally, I want to thank all the people involved in this project. So Joanna Grabowska and Martina Twila did all the mouse studies, and Alcia Avandi, together with Katarina Olesek, did all the human studies. And everything was done in close collaboration with Professor Gert Storp of the University of Utrecht. Well, thank you for your attention.
Okay, thank you very much for your presentation. We have time for one question, and one question arrived. What route of administration works best with CD129 targeted liposomes? Actually, we're now testing this. So up to now, we only have been doing intravenous administration. But you also have CD169 positive macrophages in lymph nodes. So also sub-Q administered should be able to drain to the lymph node. But whether one or the other is more efficient, we'll figure it out. Yeah. Sounds like interesting future studies will address these questions. Thank you very much. And it is my pleasure to introduce our next speaker, Dr. Yael Bavli-Felsen of the Hebrew University in Jerusalem. And Yael will present a clinical case study to share her experience with development of liposomal methylprednisolone formulation and how preclinical studies helped with translation of this formulation. Yael, please, the floor is yours.
Thank you very much. Thank you, everyone. I'm very happy for the opportunity to talk today and to present how preclinical studies helped us in the translation of our bench formulation of liposomal methylprednisolone to the clinical study that I'm going to present now. So as we saw earlier during the session, the injection of pegylated formulations can trigger a whole myriad of effects in the patient, namely infusion reactions, the activation of complement activation, which is linked, the synthesis of anti-PEG antibodies, and which can, of course, then induce the ABC phenomenon, which is accelerated blood clearance. Namely, the drug is very rapidly cleared from the bloodstream because of the antibodies. So I wanted to detail a bit more about the different reactions. Now János detailed very clearly about the infusion reactions. So I'm not going to talk too much about this. Of course, we saw that they can take a lot of different symptoms. We can have mild symptoms such as rash, mild fever, but it can go into more severe and even life-threatening symptoms such as cardiorespiratory distress. And it seems that the fastest the reaction starts after the drug is administered, and the more likely it is to be severe reaction. But since it happens usually at the first administration of the drug, it is not possible to be sure if the reaction will happen and if it's going to be severe, and we have no way to predict these reactions. For now, what we have are ways of prevent or try to prevent actually the reactions by two ways. We have premedication of the patient. We give cocktail of drugs, as we saw, H1 and H2 blockers, corticosteroids, and also a very slow infusion at the beginning of the administrations. For the first 15 minutes, we do a very slow infusion of the drugs, and if the patient hasn't developed any symptom, we can increase the rate of administration. So this is now what we are doing, but of course it's not perfect, and infusion reactions still can occur in a large variety with a large number of drugs coagulated. We also have anti-PEG antibodies. These concern mostly IgM and IgGs, and they can occur. There have been some studies showing that the administration of pegylated enzymes, notably, can increase the risk of the patient to synthesize anti-PEG antibodies, which is a problem with accelerated blood clearance. But also, several studies have found, and it's been already several years, that some part of the population without any exposure to pegylated drugs have in the blood pre-existing anti-PEG antibodies, IgG and or IgM. And this happens more and more, and this is a problem, as you can see, since people with high titers of anti-PEG antibodies can have some immune reaction, which causes the drug to be very rapidly cleared from the bloodstream. And then if the drug concentration drops very rapidly from the bloodstream, of course, we have a clear decrease in the therapeutic efficacy, which is a problem which we want to avoid. So we had doctors that called us because they had a case of a patient with intracardiac IgG flow sclerosing disease. This is a rare disease. It's actually, it can be life-threatening because the disease is consists of mass growing in the body in several organs. Now, these are not tumors. They are not composed of cancer cells. These are inflammatory cells. So surgery is not an option. First of all, because in this case, the mass was growing in the heart muscle, impairing its function, but also because since they are inflammatory cells, if the surgeon performs an ablation and left some cells in the body, they will grow and form another mass, which can be even bigger than the previous one. So treatment of choice is usually steroids, especially corticoids. And so the doctors approached us because the patient already reached the limit of toxicity for the steroids, and they didn't want any systemic administration. They wanted something that would go and act and accumulate specifically in the mass. This comes to the formulation we developed, which is NSSL MPS. This is a formulation of pegylated liposomes encapsulating the glucocorticoid methylprednisolone hemisuccinate. And this drug have been shown to be very efficacious and very safe in different models of inflammation in animals, as you can see here, the list. And the doctors also perform an angiography, as you can see here, the blood vessels, and you can see here this blush around the blood vessels, which actually means that these blood vessels are very leaky and the contrast agent leaked from the vessels in the surrounding tissue in the mass, which is actually very good for liposomes, because we know that our liposomes, and it can be shown here in an animal model, tend to accumulate in areas of inflammation, and due to EPR effect or leaky vessels, they accumulate in the areas where there are leaky vessels. So you can see here biodistribution of the drug in naive animal. They go mostly to organs, but in a model of inflammatory arthritis, you can see a very extensive accumulation and preference for the areas of inflamed tissue, here inflamed joints. So back to these adverse events that we have after the administration of pegylated drugs. Since we had done only animal experiments, we wanted to be sure to avoid as much as possible all these events that are here: infusion reactions, complement activation, anti-PEG antibody generation, and ABC. So what we did, for each step, we tried to take as much precautions as possible. Regarding the drug, we did extensive characterization of our formulation before we went to the next step. Then we performed in vitro analysis of all the factors, cytokines, factors of inflammation, activation of the complement. We also try to quantify the anti-PEG antibodies before and during the treatment in order to check that they are not being generated during the course of the treatment. And regarding the ABC, we did pharmacokinetic studies. What we did in addition, we wanted to monitor the factors of complement activation during the course of the treatment. So during the drug administration, we took blood and we checked the complement activation in vivo, not in vitro. All the details of the methods and all the results have been published in a study in Molecules at the beginning of this year. So you can see all the details there. And now we go to the characterization of our drug. So we, of course, performed all the tests such as size, drug content, zeta, potential. We checked them with a cryo-temp. We did a release test. As you can see here, with a differential calorimetry, DSC, and of course, one of the most important checks that we did was sterility and pyrogenicity, because it is crucial before IV administration. Before going to the first in human, we also did in collaboration with the NCL and Marina, we sent our formulation and we checked in the plasma of healthy donors signs of in vitro activation and other factors that could indicate immunogenicity of our formulation. So we checked in vitro activation following the IC3B marker. And you can see that a very high concentration, we have a very, very small complement activation, but this is way above the clinical dosage. So we were not really worried about this. All the other signs that we checked, platelet aggregation, hemolytic properties, leukocyte procoagulant activity, also plasma coagulation time, everything came out negative, which indicated a very low immunogenicity of our formulation. We also performed a cytokine release test in the plasma of healthy donors, because this test is very important. It is a good prediction test for cytokine release, which is very dangerous upon injection of drugs, of nano formulations, and everything came out negative. So we started to plan the, actually first in human, the compassionate use of the drug. Our formulation was injected once a week during 2 courses of treatment. The first course was 5 weeks, so 5 injections. We had a break of 6 months, then 3 weeks. We escalated the doses to go to reach 300 milligrams of MPS per week. And of course, because of ethics reasons, we had to administer premedication to the patient. So she got the cocktail of drugs aiming at reducing the risk of infusion reactions. So what we did before the drug administration, we took a blood test and we checked the presence of anti-PEG antibodies, and it was negative. So she didn't have pre-existing anti-PEG antibodies. So we started the treatment just before she received premedication. During the infusion, we took blood tests in order to check complement activation at different times. Then every 2 days, we had plasma to check the presence of anti-PEG antibodies in order to check that she was not generating anti-PEG antibodies over the days. And then as soon as it was clear that we didn't have anti-PEG antibodies, we started the second treatment 1 week after, and that was every week, so weekly treatment and blood tests. Regarding the markers of complement activation, you can see here the cascade of the complement activation. We tried to test several components, several markers. We checked C3A, BB, which is more of the alternative pathway. We checked IC3B, and of course SC5B9, the membrane attack complex, which is common to all pathways. We ended up dropping C3A because it had a lot of background, so we kept these 3 markers and added thromboxane B2, which, as János said earlier, is also a marker of complement activation. As you can see here, the results. You can see that we are below the threshold of activation during all the treatments for SC5B9. Thromboxane is also clearly below the threshold. IC3B, despite a small increase, we remained below the threshold. And the only small increase we had was in the BB marker. So during the first course of the treatment, we are here with these 3 lines below the threshold. And we started actually the second course of treatment with a small increase in BB, but we couldn't see any correlation with clinical signs. The patient didn't have any sign of infusion reaction or clinical signs of any adverse event. So this is a small subclinical increase in a BB marker, but we measured it and we saw it. We also checked for the presence of anti-PEG antibodies. So the problem is that there is no standard test for measuring and quantifying the anti-PEG antibodies. So we used the method of our collaborators in Taiwan, Steve Roffler. They sent us the antibody that they developed. It's a humanized antibody, which allows the quantification of human anti-PEG antibodies in the blood. So we collected blood from healthy donors from the university. People, students, and researchers worked with PEG because we needed to find people who are positive for IgM and IgG in order to have our positive control for the test for the patient. And among 28 donors, we found that 11 of them had IgG anti-PEG antibodies pre-existing. These people never had any treatment with PEG-related drugs. And 1 of them was positive for IgM. So we used serum, and we performed ELISA test every week before the first treatment. So as I said, she didn't have any pre-existing anti-PEG antibodies. And the first week, you can see that we are way below the threshold of the positive control for all the antibodies, IgG and IgM. And we repeated this test every week, and it always came out negative. So the patient didn't develop any anti-PEG antibody. In addition, as I said, we took the plasma of the patient and we performed a pharmacokinetic study. So we measured the NPS level in the plasma, as you can see here, during week 1, 2, and 3. You can see that the graphs are really nicely overlapping. There is no sharp decrease during week 2 or 3 of the plasmatic levels. So there is no accelerated blood clearance, and it's actually in accordance with the results of the anti-PEG antibodies that were not generated. So we're very happy. And in conclusion, we are very, very happy for the patient that she didn't develop any adverse event and any sign of infusion reaction. But we didn't see also any increase in any marker, so we couldn't do really a correlation between signs of infusion reaction and any marker that was increased during the drug infusion. But this is something that we want to implement in more drugs reaching the phase 1 or first in human stage, that we want to have more patients getting a PEG-related drug for the first time, and we want to, again, start this protocol to test the blood of the patient before the antibodies, the markers during the drug administration, in order to be able to correlate if there are any signs of infusion reactions or any side effects increase in anti-PEG antibodies, and to be able to see if we have markers that earlier were increasing and be able to detect as soon as possible in the process of the drug development, but also during the first administration, because a lot of drugs fail during this test. And as Marina said, a lot of drugs fail due to immunotoxicity and immunogenicity. So we want to be able to find markers as early as possible to detect this kind of toxicity, immunogenicity, and be able to have more drugs reaching the market more safely and to stay there for a long time. So thank you very much. These are our lab members and Dr. Kiran Turjaman here. The NSSL-MPS is really her baby. That's really a big part of her project. She did a lot of studies, and we got to the first in human, and we were very, very happy to get to this point. And of course, our collaborators, because good science is always a work of collaboration. Thank you very much.
Thank you very much, Yael, for fantastic presentation. It's exciting to see the clinical translation of the products that you are doing. We ran out of time allocated for your presentation, so we will save questions that we received for you till the end of the session. And János will present our next speaker.
Okay, the next, do you hear me?
Yeah.
Okay, the next speaker is Dr. Sarah Kraus. She is the head of biology department at New Face Ltd in Petah Tikva in Israel. And Sarah is going to talk about safety assessment of Sarah nanotechnology in swine models. So Sarah, please. Come back to the pigs.
Thank you for the introduction. I'll share my screen. Is it okay? Okay, so this is the outline of my presentation. And first I'll give a brief introduction on Siren Nanotechnology, followed by a few words on the EPR effect, swine models, Siren Nanoparticles, and the studies we have done so far in swine. So the Siren Nanotechnology, sorry. My apologies. Siren Nanotechnology is a class 3 medical device that comprises Siren nanoparticles and electromagnetic induction system. The primary indication is stage 4 small cell lung cancer, but it is also applicable for other solid tumors as well. The Siren nanoparticles, or SNPs, are given intravenously to the patient and become localized on the cancer cells through the use of the enhanced permeability and retention effect. The main innovation is the self-ability of the nanoparticle to control its temperature without inducing a thermal ablation. After administration, the patient is exposed to a low-frequency, non-ionizing electromagnetic field, which causes the nanoparticles to heat to about 50 degrees and cause hyperthermic cancer cell death at the subablative temperatures. So today I'll show you the first studies showing administration of therapeutic SNP doses, followed by EMF application in swine. The EPR effect is the central mechanism for tumor targeting. As you can see, it allows extravasation of the nanoparticles and the preferential retention of the SNPs in the tumor due to the leakage of the blood vessels in the tumor and reduced lymphatic drainage. This is a passive mechanism, and therefore there are no side effects. The Siren nanoparticles are multi-core nanoparticles that, as opposed to single nanoparticles, are able to enhance the thermic mass. Iron oxide nanoparticles, the Siren nanoparticles are composed of three main components. One of them are iron oxide nanoparticles of 25 nanometer in size that heat under EMF application. Then we have a PCM, or phase change material, that absorbs energy without heating, limits the temperature to 50 degrees, and ensures that healthy cells are not damaged. Third component is an encapsulating polymer. This is PEG 20,000 that encapsulates the PCM and the iron oxide nanoparticles, enabling biocompatibility and masking the nanoparticles from the mononuclear phagocyte system. PEG also enables flexibility in blood vessels and therefore improves the EPR effect. The total size of the SNP is about 135 nanometer, as demonstrated here by transmission electron microscopy. Swine are major animal species used in translational research, often used for surgical models as well as for procedural training. And they are increasingly being used as a good alternative as a choice for non-rodent species in preclinical toxicology testing required by regulatory agencies. Their main advantage is that they share similar anatomic physiologic characteristics to those of human, as well as comparable thermoregulatory mechanisms and electromagnetic properties, making them clinically relevant to humans, particularly in our setting. Now, I'll show you a series of studies in which we examined toxicity and biodistribution in swine, and which included the following. So first, we tested subacute systemic toxicity and biodistribution of various ACNP doses without and with and without EMF in both Sinclair and Göttingen mini pigs that were treated with single ACNP doses followed by EMF application and a follow-up period of 30 days. Then we proceeded to examine chronic systemic toxicity and biodistribution of repeated ACNP doses without EMF that were examined in a Sinclair mini pig that received 3 IV infusions at an interval of 1 month between each session. The animal was followed up for 93 days. The last study that I'll show you today is full treatment of a Sinclair mini pig that was treated with a clinical SNP dose, 156% dose, and with a follow-up period of 1 week. Now, the performance of these studies was made possible due to the upscaling of the manufacturing process that allowed us to obtain large amounts of SNPs. Before we proceed into the results, here you can see the preclinical electromagnetic induction system used in swine, which is composed of several adjunct devices. We have an RF generator that generates the electromagnetic field. Then we have an electromagnetic coil, a swine water cooling blanket system, which helps to cool the animal during the exposure. And we used fiber optic temperature probes to monitor the source temperature of the animal during the exposure. The probes were placed at specific locations around the animal. Then the animal was wrapped with the cooling blanket system and positioned in the center of the coil. One of the challenges that we encountered during development was that the system produces not only a magnetic field, but also an electric field. And this electric field penetrates normal tissue and induces eddy currents, which may result in undesired heating of normal tissue. The solution for that was the use of a cooling blanket system. And then here you can see an example of the thermal profile of an animal. In our studies, we used discontinuous irradiation that was performed by 3 intervals of 10 minutes each, with a 5 minutes breaks between each interval. And we monitor the surface temperature at the different locations. And here you can see the follow-up of the rectal or core temperature of the animal, which was lower than the normal physiological temperature. So let's see some of the results. And here you can see some of the clinical parameters that were measured during SNP administration. In the left graph, you can see measurements of the heart rate and respiration parameters that were relatively stable during the injection. And we did a full panel of hematology and chemistry parameter for all animals. And here you can see one example. This is AST, an indicator of liver function. And you can see that there were changes within normal ranges with some normal fluctuations. So SNP doses with or without EMF were all well tolerated with no adverse reactions in any of the peaks followed up for 30 days. All clinical parameters were stable throughout the administration of SNP doses, and there were no significant changes in the clinical pathology parameters, and most changes observed were all within normal ranges. Here are some of the results of the biodistribution in animals that were followed up for 26 days. And the iron content analysis was performed by using the particle electron paramagnetic resonance method that enables the quantitation of exogenous iron present in biological tissues. And you can see here that SNPs with or without EMF accumulated primarily in the liver, in the lungs, and in the spleen, with most nanoparticles found in the lungs. This is 33%. The total percentage of SNPs found in the organs was 37.3%, with approximately 63% of SNPs cleared from the body after 26 days. Histopathology analysis showed no treatment-related toxicity in any of the organs that were examined in all animals. The only finding that was observed was the minimal multifocal stained macrophages in the lungs, as you can see here in the by the green arrows. And this pigment accumulation was not associated with any inflammation, and there was no evidence for any thermal damage. Here you can see the results of repeated dose treatment without EMF in an animal that received 3 IV emissions of nanoparticles and was followed up for a period of 3 months. So here you can see the result of the iron content analysis. And again, main accumulation was found in the lungs. This is 47.8%. The total amount that accumulated in vital organs was about 52%, suggesting that 48% of the nanoparticles cleared after 93 days in this animal that received 3 repeated doses. Again, we did a blood analysis testing for all hematology and chemistry parameters. And here's one example. These are the white blood cell counts, showing again changes within normal ranges. Levels were followed up during the dosing sessions as well as the entire follow-up period. And this suggests the absence of any inflammatory reactions. Here you can see a picture of a lung section. And again, you can see pigment accumulation in the lung macrophages that was found to be positive for Prussian blue staining, consistent with the presence of iron derived from the nanoparticles. This is the last study showing a full treatment of an animal treated with the clinical SNP dose, followed by EMF application and a follow-up period of 1 week. This is toxicity testing. And the clinical, the dose calculation was based on the NOAL principle in accordance to the FDA guidelines. And the selected SNP dose used in the study represents the maximum dose that a patient would receive in a clinical trial when considering dose accumulation from repeated dosing, and actually mimics 3 consecutive doses in human. And this is 156%. And here you can see the results of the biodistribution, which were very similar to that of the animal that received 3 doses. The main accumulation in the lungs, this is 45%. The total percentage of nanoparticles found in the organs was 51.8%, with about 50% of the SNPs cleared from the body after 7 days. So to conclude, single SNP doses with or without EMF, as well as 3 repeated doses of the SNP without irradiation, all well correlated in swine models in vivo. There were no treatment-related clinical observations observed during the dosing, the exposure, or follow-up periods. There were no signs of inflammation, necrosis, or any adverse reactions, including kappa, suggesting that the SNPs do not degrade, cause toxicity, or any thermal damage, even after 3 repeated doses of SNPs or a clinical dose of 156%. Most of the nanoparticles clear from the body within the first week after the injection. Overall, and these assessments open new avenues for generating Siren Nanotechnology as a potential novel therapeutic modality for the treatment of cancer patients. And a few words on our future plans. Here you can see the clinical system to be used in humans. And we plan to complete a GLP study in Sinclair mini pigs in the coming month. We also plan to start a first-in-human clinical trial next year. And I would like to thank the summit organizers for the opportunity to present our data, our new phase team, and all the people and organizations that took part in these challenging studies. Thank you for your attention.
Thank you very much, Sarah, for the presentation. Again, we will keep the questions until the end of the session. And it is my pleasure now to introduce our next speaker, Dr. Kirill Afonin of the University of North Carolina in Charlotte, will talk about immunomodulation with nucleic acid nanoparticles.
Thank you very much, Marina. And I would like to thank the organizers also for providing the opportunity to share our research. All right, so thank you again.
Yes, your presentation.
Excellent, thank you very much. So, as we all know, RNA and DNA are integral biomolecules, essential for force of life. And if we look at the central dogma of molecular biology, DNA goes to RNA to protein. Only 2%, about 2% of DNA actually is being translated to protein. However, about 60% of our genome is being transcribed into DNA but not translated. And the question is, which molecule of our life, of our body, determines the complexity? And if we look at the proteins, we express about the same number of proteins as other organisms, and some organisms such as this flower, for example, Arabidopsis beats it expresses more proteins. So in fact, our complexity is determined by the RNA, which is being transcribed but not translated, as can be indicated by this nice graph, where you basically plot non-coding DNA over the total genomic DNA. You can see that this ratio increases with the complexity of the organism. And we know that RNA now is responsible for orchestration of many cellular processes along the central dogma of molecular biology, and as such, it becomes a very potent therapeutic target, and in fact, it can act as the therapeutic itself. And recent clinical success of multiple therapeutic nucleic acids and the very exciting approvals by the FDA of 2 RNAi-based therapeutics, patisiran and givosiran, have simulated rapid transition of nucleic acid nanotechnology towards their therapeutic application. So we know that RNA can act as a functional material, so it can be a therapeutic nucleic acid by itself. It can regulate gene expression. It can participate in genome editing, act as the catalyst, and so forth, so forth, so forth. So these classes can become potentially therapeutic nucleic acids. So the question is, what if we want to deliver therapeutic nucleic acids of different kind to the same cell how do we guarantee that every cell will get exactly the same cocktail and composition of therapeutic nucleic acids? That's where the other property of RNA comes into the play, which is acting as the scaffold, a neural scaffold. And basically what our lab does is that learn from nature, we build artificial small nucleic acid nanoparticles, mostly made of RNA, and then decorate them with different therapeutic nucleic acids. So these are atomic force microscopy images of different shapes. We can do 2-dimensional, 3-dimensional shapes, fiber shapes, and we can decorate them with different number of therapeutic nucleic acids, for example, siRNAs or microRNAs or aptamers and so forth. And we can trace their behavior in silico, and actually this technology has been licensed by Sixfold, where they use some of our constructs for drug delivery. So how do we program this? Again, we learn from nature. We look at the biological molecules, natural molecules, RNA molecules. We see how the folding provides a particular function that they obtain upon the folding. We can also implement some in silico design. We can do SELEX, which is in vitro selection of RNA or DNA molecules with a particular function. And then basically by looking through the resonance structures, we identify sets of secondary structures of RNA or DNA, which position themselves in a specific way in the 3D space and gain particular geometric or interacting properties. For example, we can identify a structure which will guarantee 90 degrees angle between two adjacent helices, okay, as the king term. And we call those fragments of RNA with a particular structure or interacting properties motifs. And then we basically use them as Lego blocks and put them together in different orientations and different order to make nucleic acid nanoparticles. And this can be done by hand, design, or we can use some computer assisted approaches. For example, one of the programs is Nanotyler, which would identify structural or interacting motifs, and then through the series of optimizations would identify optimal geometrical parameters required for formation of particular nano shape. In this case, it's a ring. And as such, we can create a large library of nucleic acid nanoparticles of different shapes, compositions, physicochemical properties, and so forth. But the next logical step would be identify the immunostimulatory properties of these particles, since we intend their use in vivo and essentially as therapeutics. So we need to understand how they behave with the human immune system. So literally, we have hundreds of nucleic acid nanoparticles currently available to the researchers, and they all have sophisticated shapes, different design principles, different assembly approaches, and so forth. But what unites them all is pretty much unknown immunostimulation and unknown interaction with the immune system. So our team, in collaboration with Dr. Dobrovolskaia a few years ago, asked those important questions: how nucleic acid nanoparticles can be recognized by the human immune system, and whether or not a particular shape and conformation and size of nucleic acid nanoparticles can induce a specific type and the level of a given cytokine response. Again, this work that I'm presenting today was done in collaboration with Dr. Dobrovolskaia. We spent maybe like 5 or 6 years of hard work. We published quite a few papers on that. And basically what it allows to do is to come up with a very standard protocol, experimental procedure that details the preparation of endotoxin-free, pyrogen-free nucleic acid nanoparticles, their characterization, also the assessment of the immunological properties in human peripheral blood mononuclear cells, PBMC. So this protocol also details all the essential steps of isolation of PBMC, and then the treatment with different nucleic acid nanoparticles. We also suggested the controls that should be used, which give reproducibly relevant results. And this analysis is done for characteristic biomarkers, which were chosen to be type 1 and type 3 interferon, which showed to be with the least interdonor variability among other cytokines that were tested, and we tested more than 30 different cytokines. So this procedure, the continuous execution of this protocol wouldn't exceed 48 hours, and if you don't have a possibility of immediate analysis, single use aliquots of supernatants collected from PBMCs can be frozen and stored and analyzed later, or can be shipped for analysis somewhere else. So this procedure was recently published in Nature Protocols, and we really anticipate that it will be of great interest to the major participants in the field of nucleic acid and therapeutic nucleic acid technologies, and will be used as the gold standard for their characterization. So basically, if you follow these protocols, you can see very characteristic responses for different types of nucleic acid nanoparticles, whether they're globular or planar or fibrous, whether they're functionalized extensively or moderately. It can basically, the system can pick up the subtle differences in nucleic acid nanoparticles in the form of the interferon response, type 1 and type 3 interferon response. So right now, for the sake of time, I will just give you the quick summary of the design rules that were identified to be responsible for the immune recognition of nucleic acid nanoparticles. So we identified that dimensionality of nucleic acid nanoparticles is extremely important. And if you go from globular to fibrous, the interferon production drops. The composition is important for a given nucleic acid nanoparticle of this particular sequence and size. DNA versus RNA produce different levels of response, with DNA nanoparticles being immunoquiescent, while RNA would be immunostimulatory. Functionalization on average increases the immune recognition of nucleic acid nanoparticles. So basically, you take any of the previously characterized scaffolds and then extensively functionalize them, and they will all produce comparable numbers of interferon productions, despite of the fact that individual scaffolds can produce different levels of interferons. However, the extent and dilatation of functionalization of therapeutic nucleic acid nanoparticles additionally can tune the immune response. We didn't find much response from the sequence of nucleic acid nanoparticles, meaning that the same exact topology can be obtained by using a different set of RNA or DNA sequences. And in this case, the sequences do not determine the immune response, but rather shape only. We identified that without carrier, all nucleic acid nanoparticles are immunoquiescent, and as such, they can be easily used for safe extracellular activities. However, in the presence of carrier, plasmacytoid dendritic cells become the main responders to the interferon production in response to different nucleic acid nanoparticles analyzed. However, if you remove PDCs, plasmacytoid dendritic cells from PBMC, then subpopulations of other cells can differentiate between different shapes, and the levels, basically quality and quantity of cytokine production varies depending on the shape. We were also the first to show the machine learning approach in collaboration with Dr. Zakharov, where we could analyze a large set of nucleic acid nanoparticles using their physicochemical parameters and sequences as descriptors, and we could identify the main criteria responsible for immunostimulation of nucleic acid nanoparticles. This criteria are listed here. All this data is published in our recent review and multiple other papers. We also identify several cellular mechanisms responsible for production of inflammatory cytokines and interferons. And basically, depending on the carrier and depending on the nucleic acid nanoparticle, we can activate or deactivate certain pathways. For example, we developed a system which allows us to activate RNA interference inside the cells, but also activate DNA sequences, which we call DNA decoys and FKB decoys, which would bind activated in FKB in the cytoplasm and prevent it from entering into the nucleus. So, as such, we can not only activate therapeutic modality, but also deactivate undesirable production of pro-inflammatory cytokines. Different endocytic and cytoplasmic pattern recognition receptors can be activated depending on the nature of the carrier, and more work is being done right now on these mechanistic studies. So overall, what we came up with is that we're developing now a new platform, Smart NAMs. This name was suggested by Dr. Dobrovolskaia, specific modular, adjustable, reproducible, and targeted nucleic acid nanoparticles platform. And we envision it to be a very versatile platform intended for intracellular use as well as extracellular use, because remember, I told you that our nanoparticles without use of any delivery carrier are completely immunoquiescent, and as such, they can really perform the function comparable to the function of monoclonal antibodies and so forth, but they can also elicit particular immune responses beneficial for particular treatments and immunotherapies inside the cells once they deliver with the proper carrier. With that, I would like to thank all the collaborators which participated in this project, especially Dr. Dobrovolskaia again, because we pretty much did all this work together, my lab, and if you have any questions, or if you want to read more about some news about this research, please visit our website. And thank you for your time.
So I will take a pleasure of introducing our next speaker, Dr. Adelina Heller of the Max Planck Institute, and she will talk about the effects of heparin on the cellular uptake of nanocarriers. Adelina, please, the floor is yours.
Thank you very much for the nice introduction. I'm going to present how heparin affects cellular uptake of nanocarriers. Heparin is a polyanionic sulfated glycosaminoglycan with a strong binding capacity to antithrombin 3. In clinic, heparin is widely used for thrombosis prophylaxis at low doses subcutaneously, but also intravenously at high doses ranging until 7.5 units per milliliter in cardiovascular interventions. The study I'm going to present bases on the findings of Carol Champagnat, implementation of the negatively charged heparin molecule into the protein corona of nanocarriers causes changes of uptake in the phagocyte cells. The extent and direction of uptake depends on cell type and concentration of heparin. For more information on this finding, you can have a look at our poster. Basing on the finding that heparin-coated nanoparticles modulate the uptake in the phagocyte cells, we want to investigate if co-medicated heparin has potential to modify pharmacokinetics of nanotherapies. Topic 1 will be how presence of heparin favors the cellular distribution of an antigen shell nanovaccine towards dendritic cells. In the second topic, I will talk about the effect of an intravenous heparinization on biodistribution of liposomes. The pharmacoefficiency of nanocarriers is dependent on cellular uptake of the target cells, which can be dendritic cells in case of nanovaccines or tumor cells in case of liposomes loaded with anti-tumor drugs. The cellular uptake in turn is dependent on blood circulation of nanocarriers, and blood circulation is dependent on the clearance via the reticuloendothelial system, RES, which consists of spleen and liver, and the local phagocyte cells, as we've learned already from Joke. In this study, we investigate if heparin alters the accumulation in spleen and liver, and if heparin thereby alters the cellular uptake of local phagocyte cells, which are responsible for the blood clearance of nanoparticles. I will now start presenting topic 1, how heparin favors cellular distribution of an antigen shell nanovaccine towards dendritic cells. The nanovaccine in question is a protein capsule with shell is constituted of the model antigen ovalbumin. When the nanovaccine is taken up by dendritic cells, the shell is digested by proteases and antigen fragments are cross-presented by the activated dendritic cells. In vivo and also in vivo, in vivo and also in the here shown experimental setup, dendritic cells compete with macrophages of the spleens for uptake. The effect of heparin on the uptake by competing phagocytes and on dendritic cells was investigated by incubating a spleen cell mixture with heparin and fluorescently labeled nanocapsules. For investigation, the effect of heparin, the medium was supplemented with clinical relevant concentrations of heparin ranging from 0.25 to 4 units per milliliter. Here, the normalized uptake of capsules towards the non-heparin-treated control on the very left is presented. This graph shows a significant reduction of uptake of around 50% for the three main populations of splenic macrophages. Having a look at the dendritic cells, the high-dose heparin-treated group on the very right did not differ significantly from the untreated group. This experiment shows that heparin causes a significant reduction of uptake into RES macrophages, but not into dendritic cells. Now we want to investigate if a high-dose intravenous heparinization causes this favorable effect towards dendritic cells for the ovalbumin nanocapsules also in vivo. Therefore, bulb C mice were heparinized, aiming a final blood concentration of 4 units per milliliter, 15 minutes prior to injection of the Cy5 labeled ovalbumin capsules. Following, biodistribution and cellular uptake were assessed after 30 minutes and 6 hours. The emission of Cy5 was measured for the organs. At 30 minutes, here shown, a significantly higher accumulation was observed in the liver, but slightly higher for the heparin-treated group, which is blue. And after 360 minutes, the slight but significant effect was equilibrated. In contrast to the significant changes we've seen in vitro, in vivo, no significant alteration of cellular uptake into the RES macrophages was observed. Summarizing topic 1, we have shown that the presence of heparin causes a reduction of uptake into RES macrophages, but not dendritic cells in vitro. An intravenous heparinization in vivo caused a slight increase of ovalbumin nanocapsules in the liver, but in contrast to in vitro, no alteration of cellular uptake. We presume a decrease of the blood heparin level by the biological clearance mechanism falling below a level where the uptake reduction effect observed at 4 units per milliliter does not occur anymore. And we conclude, nevertheless, in this study, heparin emerged as an interesting molecule for passively targeting dendritic cells and thereby as a modulator for protein-based antigen shell nanovaccines. The second topic I'm going to present is about the effect of an intravenous heparinization on biodistribution of liposomes. By now, since the approval of Doxil, liposomes have become broadly applied drugs, not only in cancer therapy, but also in fungal diseases, analgesia, and vaccination as well. The chance is quite high that patients undergo a heparinization while liposomal therapy. Our question is, if heparin influences the pharmacokinetics of liposomal therapies? If yes, it would be of concern for design of many clinical medications. For the following investigations, we focused on the formulations of Doxil and Ambisome. As shown before, the same in vitro setup was carried out to investigate the uptake of liposomes while heparinization. Clinically relevant concentration between 0 and 4 units per milliliter were applied. The liposomal formulations were additional DID labeled, and in case of Doxil, where the active substance is normally encapsulated and not drug-loaded for this study. For the 3 macrophage populations of the spleen we have investigated, we see a different behavior of uptake and presence of heparin. While the highly negatively charged formulation of Ambisome, green, tends to rise uptake and presence of heparin, the low negatively charged formulation of Doxil decreases its uptake into the macrophage populations of the spleen and presence of heparin. At a high dose of 4 units per milliliter, 4 units per milliliter in the medium, this decrease is significant for the red pulp macrophages. After having shown changes of uptake at a high dose of heparin, which was significant for the Doxil formulation, the formulation, we now want to investigate the effect of a heparinization on biodistribution and cellular uptake in vivo. Therefore, we work on with the formulation of Doxil, which is also a broadly used therapeutic in clinics. Yeah, investigating biodistribution and cellular uptake of the Doxil formulation while a high-dose heparinization animal were intravenously heparinized to a final blood concentration of 4 units per milliliter, as described before. And fluorescent in vivo imaging showed an initially increased accumulation in the spleen for the heparin-treated group, which is blue, and also in the liver, a significant increase of the liposomes, of the liposomal accumulation was observed for the heparin-treated group, which is blue. And at 6 hours, the differences were equilibrated. Yet the organ fluorescence distribution shows that the spleen harbors the highest density of the liposomal formulation. The accumulation for the heparin-treated group, which is blue, is significantly stronger than for the PBS-treated group in the spleen at 30 minutes and 120 minutes. And also in the liver, we see this significant increase of accumulation at 120 minutes. At 6 hours, the differences in liver and spleen are equilibrated. Flow cytometry analysis on the macrophage populations of the spleen revealed no difference of uptake for the heparin-treated group towards the PBS-treated group, neither at 30 minutes nor at 360 minutes. So, in vivo, a high-dose heparinization caused an increased accumulation in the liver and spleen within the first 2 hours. No change of uptake into the red pulp macrophages, white pulp macrophages, and marginal zone macrophages of the reticular endothelial system was observed. To summarize topic 2, the presence of heparin influences liposomal uptake into RES cells in vitro. A heparinization influences biodistribution of liposomes, but not cellular uptake in vivo. And we conclude a high-dose intravenous heparinization slightly but significantly alters pharmacokinetics of liposomes. But we do not expect relevant effects on pharmacodynamics by this slight but significant initial increase of the biodistribution in spleen and liver. And I prepared 3 take-home messages. So in vitro, the presence of heparin alters cellular uptake in the phagocytes of the spleen. In vivo, a high-dose heparinization leads to an initially increased accumulation of nanocarrier in the spleen and liver. And in vivo, a high-dose heparinization does not cause alterations of cellular uptake in the phagocyte cells of the RES. I want to acknowledge my 2 groups at the Department of the Dermatology and at the Max Planck Institute for Polymer Research, to my supervisors, Volker Mailänder and Katharina Landfester, and to the collaborators of the Department of the Pharmacy. And of course, to the Collaborative Research Center 1066, of which my project is part of. Thank you very much for listening and watching.
Thank you very much, Adelina, for the beautiful presentation. János and I would like to cordially thank all the speakers and all audience that joined this session this morning. We have few minutes to ask questions to the speakers, but I was informed that this session will automatically terminate in 4 minutes. So János and I invite you to join the speakers at the debate launch 2. In order to get into that debate launch, you go to the clean them, just a regular meeting, join live stream, and then at the bottom of the screen, you should see 3 buttons that would take you to the appropriate debate launch. So we will be at the debate launch 2. And now in the remaining minutes, we received several questions. The first question is for Yael. The question is, in your study of the anti-PEG antibodies in healthy donors, what isotype of IgG did you detect? Was it IgG1, IgG2, IgG3, 4? Could you please elaborate?
So we did a very quick study of the to detect the anti-PEG antibodies. We didn't go into the subtypes because that was not the point of the study. We just wanted to get some positive control, but we definitely need to go, first of all, get more donors and to have a bigger study and to go into the subtypes. I'm pretty sure that Steve Roffler in Taiwan checked the subtypes that they got, but we didn't check.
Okay, thank you, Yael. Thank you. The next question is to Kirill. In your studies, then you make DNA nanoparticle. What is the backbone of the DNA that you use to construct your nanoparticles? And if you think that the type of the backbone would influence the immunotoxicity or immunological properties of these particles?
Right, so we only use the natural backbone, so phosphate backbone, but we also tried recently some chemical modifications, which were introduced mostly at the sugar side, so at 2' side of the sugar of the ribose, we tried all methyls, we tried fluorinated groups, and basically all these modifications allow us to further tune the recognition of our nanoparticles by different PRRs, pattern recognition receptors. For example, some of these modifications would promote the recognition by toll-like receptors, but the others, if you take exactly the same particle but introduce instead of OH group, for example, fluorination or O-methyl, will abolish this recognition, but rather promote the cytoplasmic recognition of the nanoparticles by the RIGI, for example, or POL3. So we have recently published a work on nucleic acids research, actually last week it was accepted, where we showed that by simply changing between DNA, RNA, and 2' fluorinated RNA strands, but using the same connectivity of nanoparticle, you can tune precisely the route of recognition and the extent of the cytokine production and response of nanoparticles. But that's an excellent question. Thank you.
Thank you. So we also received questions for Sarah and János, but since it is 10:15 in Frederick, Maryland, and it's 15:15 in Basel, we would like to thank one more time all speakers and all audience for your attention, and speakers for beautiful presentations, and please connect to the debate launch to continue this discussion. So we'll see each other in a few minutes. Thank you, everyone.